Dear community, this is the big Nobel Laureate speech of this CLINAM. And Susumu-san, I am so glad that you came. You have a very full program. It's hard to get you. Usually I can only speak to your assistant. And it's very, very seldom that you come yourself to the phone. You are coming from MIT to us. But let me tell them to the people that after your first degree in Kyoto, you already went to the United States, where you were living in San Diego, and then you got your PhD there. And from there, your traveling was quite directly to Basel. And now I will continue in Schweizerdeutsch, because I know that you speak so well and understand all the words in Swiss German. Basel is not unknown to you since you have been living 10 years here and you worked at the Institute of Immunology of Hoffmann-La Roche. Actually, I was told recently that you liked this time in Switzerland, you estimated it, but now you are back in America and you are at MIT. Basel is a significant landmark in your life for your research of using advanced techniques of gene manipulation. You have received in 1987, the Nobel Prize for Physiology. Today, your research of excellence has changed to the field of neuroscience, and we look very much forward to your intervention on learning and memory. Thank you. The floor is yours.
I hope the microphone is on. Can you hear me? Okay. Good morning. So thank you for this wonderful invitation. It looks like a very, very interesting conference. As Dr. Löffler told you, Basel is my second hometown. I stayed here 10 years in the 70s, and we had nothing but wonderful, I have nothing but wonderful memory of my stay during the time of my stay in Basel. So today, I'm going to talk about some brain mechanisms underlying memory. Okay, I can tell you just briefly that during the past 3 to 4 years, I would say there has been a revolution in the memory research. And some of that I will try to briefly summarize for you. Why do we study memory? Because memory is a major cognitive functions evolved along mammals, particularly for human beings. Memory is not just the favorable episode you may have formed a memory when you were a child, or maybe some memory for the missing bunch of kids, like shown in this cartoon, the picture. But memory is basically all the information you have acquired up to this point, since you were born, through your series of experiences, episodes. Okay, so apart from genes you are born with, the memory, which information obtained by memory through your experience of numerous events, is the source of your self, your personality. Okay, this is all the human cognitive functions, other things like emotions and decision-making, consciousness, all these things is linked to the memory information you stored in your brain network. And as you know, if we lucky and unlucky losing this memory system, then you'll have this very serious problem of a dementia, like Alzheimer's, who would when a disease advances, very difficult for them to behave like usual human beings. So this idea, memory is actually stored in some kind of a physical changes in the brain network, goes back at least to Greek time. The Plato has already mentioned this idea. Okay, but it took centuries for this idea to be more scientifically formulated. And as shown on this slide, Richard Zeman, German psychologist, published this book called Die Mneme, in which he coined the word memory engram. Okay, so and then he theorized how this memory is stored as a physical alteration in the brain network. But his idea was completely ignored by people around that time, and since after that, until 1970, late 70s, early 80s, this guy, Daniel Schachter, who is currently the chairman of the psychology department at Harvard University, published this book and tried to revive Siemens' engram theory of memory. Okay? And now, in the middle of last century, also the person who had a tremendous impact in the memory research is this man, Donald Hebb, a Canadian psychologist. And he had this idea of neuron synapse changes in synaptic efficacy. It's called synaptic plasticity. Is a major molecular or physical basis for memory information storage. Now, since based on a variety of studies which define the neurons, synapses, neuronal circuits, and so on, the following definition could be formulated about episode and cells, neurons, which contain the memory engrams. Okay, so according to this idea, and this is for most common form of memory called episodic memory. That is the memory of events that occurs to you, okay, episodes. So I just read this. Certain population of neurons, which will be named as engram cells, encode information extracted from an episode by being activated. Activated neuron means cell fires, okay? And undergoing enduring, lasting physical and or chemical changes, which is called engram, as the Zeman defined. Subsequent, the next sentence is very important. Subsequent reactivation of these cells by stimuli that are part of the original set of encoded stimuli results in the phenomena we call recall, and a recall of that particular targeted original memory. So for certain population of cells or certain changes that occur in the brain by the process of so-called learning, in order for them to be called engram cells or engram themselves, these conditions has to be met. And this has been very difficult, mainly because of insufficiency of technology. Until about 5 years ago, the new technology was invented by combination of several different kinds, and this has become possible. Now, in order to prove the certain population of cells are engram, memory engram-bearing cells, the brain researchers have expressed the conditions by which this can be accomplished. Okay? So here's one example. This is Martin and Morris published in 2002 in one of the reviews. So it's already a dozen years ago. So how to demonstrate the memory engram? The final test of any hypothesis concerning the memory engrams must be a mimicry experiment. You mimic the experiment. You mimic what may be happening in the brain of live animals. The mimicry experiment, in which apparent memory is generated and expressed artificially without usual requirement of sensory experience. So you take away sensory experience, and yet the animal behave as if they form the memory and recall the memory. That's the kind of thing one has to do in order to be able to call certain population of cells, memory engram cells, and then these changes you can find in those cells are memory engrams. So people in my lab, several people, launched this challenge, and using as I said, combining several technology. And here the three important tools were used. The first is the method to label the neurons activated by learning. So that was a part of the condition for memory engram. So for this, we use something called immediate early gene promoter, called the C-force promoter. And the second is the method to target this labeling to the specific time window of the encoding of the specific memory. Okay? And for this, we use something called doxycycline induction system. And the third, and probably most important technology, was that the method to directly reactivate the specifically labeled neurons subsequently in order to let animal to show behavioral memory recall. And for this, we use this opsin called channel rhodopsin. For the behavior, by the way, we all do this work in mouse, not in humans. Okay? And for the behavior, learning behavior, we use something called the contextual field conditioning, which is the rodent version of Pavlov classical conditioning. You put the mouse in a chamber with very specific feature. Mouse will go explore the chamber and acquire the memory of the feature of that particular chamber as opposed to other chambers. And then you give them electric shock to the footpad. The animal immediately form a memory of this chamber is dangerous place. Okay? And this can be tested the next day by putting a mouse back to that same chamber, and then the animal will show their memory recall of this chamber is dangerous memory by freezing, not moving. Okay? If you put the animal in another very different chamber, as shown in here, and then this will not induce the recall of the fear memory, and therefore animal will not freeze. Okay? So this system is very well established among memory researchers. And then this is the fundamental protocol for identifying specific memory engram cells and reactivating them with artificially applied delivered light. Okay? So what you do is you start with this transgenic mouse in which C-force, immediate early gene promoter, derived the expression of a transcription factor called tetracycline transactivator. So they start with a transgenic mouse. To this mouse, to the brain of this mouse, in fact, specifically to area of the brain called the hippocampus, and further specifically in the area called, the sub-area called the dentate gyrus, you target the injection of a virus which carries the channel rhodopsin gene combined with a fluorescent marker called EYFP, and also targeted to the dentate gyrus here is micro-optic fibers through which we can deliver this light. Okay. Now, animals are grown under the condition in context, specific context, under the condition where the cell would, the shear force gene will not be activated because of the presence of the recycling in the diet. And then on the day of experiments, you transfer animal from context A to very different context B. You start the experiment. At the same time, you open up window for labeling the potential engram cells with channel rhodopsin by shifting the diet from DOX plus to DOX minus. Okay? So cells which are activated by training, in this case, the delivery of a foot shock, will be labeled with a channel rhodopsin because the shear force gene will be activated and the channel rhodopsin is under the control of shear force promoter. Okay? Now, next day, you put the animal back to the context A, which is different from context B, and therefore, by itself, animal will not freeze because context A is a long chamber. They were never shocked in the context A, so they will not freeze. They will just move around. Okay. However, if my postdoc turn on the light, blue light, and the blue light is delivered to the dentate gyrus of hippocampus and activate the cell population that were activated by learning, then animal will show freezing, indicating they are recalling the memory, just being directly artificially activated in the hippocampal dentate gyrus areas. Okay? So there are a lot of control experiments, but I will not describe this. This was published already in 2012. If you are interested, I suggest you to look at it's the Nature paper. But anyway, so this allowed us to, and this is the result we obtained, by the way. This is the test day in day 3. And then if there's no light, blue light is delivered, animals do not freeze. This is the freezing in the y-axis. If you turn on the light, animal will freeze, showing a recall of the memory. And then if you turn off, this is reversible. Then they stop freezing and moving around. And you turn on the light again, and then they stop moving around. So from this fundamental experiment, very basic experiment, we could already conclude, as shown in here, immediate early gene shear force combined with optogenetics can tag or label neurons in the dentate gyrus granule cells that are activated during the memory encoding for later manipulation. Okay? And also artificial light reactivation of these memory engram, putative memory engram bearing neuron is sufficient for behavioral memory recall for the contextual fear conditioning. Okay. So this satisfies the requirement of calling these cells being engram cells. And there is one thing is missing still relative to the definition I gave you. That is, what kind of enduring changes have occurred in these cells as opposed to other neighboring cells. Okay? So this was investigated more recently. And what was expected is that these cells undergo some changes in the synaptic strengths a la Donald Hebb. Okay? So we wanted to test this. So in order to do that, we labeled dentate gyrus engram cells again, and then at the same time label upstream internal cortical cells with a channel rhodopsin. So here and then you, after 1 day of so-called consolidation window is provided, then animal is killed, and you make a slice of hippocampus, and this is the dentate gyrus granular cells here, 1 star and 2 stars. And by the presence or absence of M-Cherry fluorescence, you can tell this cell, 1 star cell, is non-engram holding cells. And the 2 star cell right next, close to it, is actually M-Cherry positive and holding this specific memory information. Okay, so you can poke those cells with so-called patch clamping technology and see whether synaptic strength is higher in engram positive cell versus engram negative cell, and that's exactly what was found here. Engram positive cell have stronger synaptic connection compared to the engram negative cell in the same animal on the same slice of the hippocampus. Okay, and now if you look at also another way to look at the synaptic strength efficacy is by scoring the density of spines, which is shown in here. And again, engram positive cells showed greater, significantly greater spine density compared to engram negative cells. Okay. Now, what is amazingly interesting is that if you add the protein synthesis inhibitor right after training, right after the shock, electric shock delivery, it's been known this will cause amnesia. In other words, in 24 hours later, animals are supposed to not be able to retain the memory because new round of protein synthesis is supposed to be necessary for the memory engram cell to get stabilized. Okay? Sure enough, when we looked at another group of mouse which are treated with azithromycin, then in the engram positive cell were not particularly more potent in terms of synaptic strength compared to engram negative cell. And the same thing is found in the spine density. There are no effect, no differences of positive and negative cells. Okay. So this is what we would have expected from what the people had, memory research had known. But it was the first time this was demonstrated specifically using the engram positive label cell and the negative cell by comparing in a single animal and a single brain. Now, surprise came when this animal was tested by behavior. Okay, so again, we went through this protocol of labeling engram cells, and also as the animal goes through the context where conditioning, and sure enough, after 1 day later after conditioning, if you look at the memory using this natural recall cues, as you can see here, animal is amnesiac. They can't remember very well. They don't freeze with the natural cues. The surprise is, if you, the next day, if you try to make animal artificially recall memory, as indicated here, right off, right on, off, on again, then azithromycin-treated animals who are supposed to be, who are amnesiac by natural cues, actually exhibit memory as much as the normal animals which have never been treated with protein synthesis inhibitor, indicating although these animals show amnesia, in fact, this cell population, which is labeled with the channel rhodopsin right after by training, it actually contain memory information. Okay? So otherwise, they will not freeze when these cells, population of cells are activated. So that was a little bit of surprise. So we have to change a variety of conditions for doing this memory, the behavioral test, and we are convinced this is true. This is not an artifact. Okay. So now the big question is, if Hughes claim the synaptic plasticity, increased synaptic strength, is not the source of memory information holding, what is underlying memory information? Okay, so our idea we tested is it is not individual synapse changes that underlie memory information holding, but it's a connectivity of memory engram cell populations. Okay, so to test that idea, we again went back to this labeling of engram cells in the dentate gyrus as well as downstream of dentate gyrus, which is called the CA3 areas. So both are labeled now, and then after treatment with anisomycin or without anisomycin, 1 day later, you look at the connectivity of engram cells downstream CA3 cells, specifically with engram cell upstream in the dentate gyrus. And this is exactly what we found. The engram cell downstream is preferentially connected to the engram cell upstream in the dentate gyrus. And no engram cell in the CA3 had much poorer connection to the upstream engram cells. An amazing thing is this specific connectivity is not affected by anisomycin treatment. Okay, even when animal are amnesiac, as revealed by their natural cue mediated record, even then, actually specific connectivity of engram cells are maintained. So we come to the conclusion that connectivity of engram cells along the pathway of information transfer is the basic mechanism for memory information storage and retention. So up to this point, the conclusion is engram cell ensemble for a given memory are organized in an engram-specific cell pathway, and the preferential connectivity between engram cell ensemble along the engram cell pathway may be a crucial substrate for storage of memory. Neither LLTC type synaptic plasticity nor structure spine plasticity is necessary for the retention of a consolidated memory. These types of plasticity may instead be necessary for memory retrieval. Our data support the retrieval rather than storage theory of amnesia. Okay, so now I think I'm going a little bit, taking a little bit too much, so I want to skip a few slides, and I just want to let you know that actually the episodic memory often comes with what is called valence, positive or negative valence. You had a great time and you formed a positive valence memory, or you had a terrible time like this fear conditioning and therefore had a negative memory engram. And our data, which I skipped, indicated that these pathways which hold the positive memory, positive valence memory and the negative valence memory actually compete inside the brain. Okay, so depending on which one is stronger, the final result valence could be positive or negative. So we wanted to find out whether this finding could be used for more translational purpose. In other words, to try to see whether by manipulating engram cells artificially with optogenetics, we wondered whether the mouse model of depression can be cured. Okay, and for this purpose, we subject mouse to chronic stress, and chronic stress is known to cause at least two types of impairment, behavioral impairment. One is this depression-like phenotype, indicated by here. And there are two different tests for this. One is called the tail suspension test, and the other is for sucrose preference test. And this is the test of ability of the depressed mice or depressed mice to cope with a difficult situation. Okay, and this is the the human depression patient exhibit similar kind of disorders. And the other one, the other one is the sucrose preference test. Is for test of whether the patient subject wants to enjoy some present experience. Okay, so anyway, so these animals also showed some anxiety problem. So now remember the purpose of this experiment to see whether if animal receive, I'll try to finish soon, the positive memory, would they, before they get stressed and fall into depression, and if you stimulate those engram cells, positive memory engram cells, would it reduce the depression-like behavior? Okay, and for that purpose, it's a complicated slide, but focus on this first one here, orange one here. So animal, this is the male mice, and the male mice is allowed to spend the time with a female mice for about an hour, which is a very robust positive memory experience for male mice. And then animal goes through the chronic stress treatment, that and then they fall to the depression state. Okay, now you test this animal and compare with a variety of control animal, and here is the final result. This is tail suspension test, and here the light off and the light on. Blue light it was turned off and on while test is being conducted, and you can see here this green bars, green lines is the only one which improved the symptom as a result of the activation, light activation of positive engram cells. Okay, and the same is here in sucrose test. Okay, so it looks like our hypothesis worked, and in fact, this slide shows that the circuit from hippocampus to an area called the BLA, amygdala, and the further downstream of nucleus accumbens cell, that process information of positive memory information is crucial for this rescue of depression type behavior. And finally, we have also shown that this effect of positive memory activation lasts beyond the period where the cells are being activated with light. Okay, so we could do it like 2 days after those cells are activated multiple times with light artificially, and then you test 2 days later, test animals' behavior, and we can see here clearly to compare the control animal, that animal improves their behavior. So that's all I want to say, and quickly summarize this. First of all, number one is dentate gyrus granular cell population identified by the C-force channel adoption labeling technology fulfills the multiple criteria for memory engram cells. Okay, and then the 4 criteria are listed here. Okay, but I will skip that. And then among the enduring changes, late LTP type synaptic plasticity is not necessary for memory retention, but seems important for memory retrieval. For each memory, multiple engram cell population form engram cell pathway from dentate gyrus, the hippocampus to amygdala to dentate gyrus in the case I described. The learning-dependent establishment of a preferential connectivity between multiple engram cell population along this pathway seem to be important for memory retention. And finally, this is probably most relevant for the theme of this conference, that is artificial manipulation engram cells allow an inception of false memory, which I did not talk about, a switch of memory valence I briefly mentioned, and an attenuation of depression behavior in mice, providing implication for future development of new synaptic methods based on the direct manipulation of neuronal ensembles, okay, which is not necessarily depend on the drugs, but by the physical method. Okay, so we can discuss this possibility for in a minute. I just want to acknowledge people who contribute to this work, and because of the time shortage, I will just feature one person, the Shuru, on top here. He's been my postdoc for the last 5 years, and he was one who really invented this technology of identifying and manipulating memory engram cells in mice, okay? Unfortunately, he died a few months ago, okay, very prematurely. So we are very sad about this. But anyway, this is what I wanted to present to you, and I hope we have at least 10 minutes or so to do the following discussion. Thank you very much.
Thank you very much.
Yeah, sorry to take it.
We sit here.
Yeah, we sit there. Okay.
Thank you, Susumu-san. We go for questions or comments. Yes, please, Rutledge.
Susumu, great talk. I had 2 questions. The first one is, as you know, in depression, it usually recurs over time. Are you able to see whether or not there's an increase in fiber connectivity to the nucleus accumbens? And does that tend to stay or decrease? That's the first part.
That, you know, you're talking about the human, okay? And then you have to remember, this is all work in the mouse, and you can't really translate it directly to human, okay? But yes, we could in a mouse model. I mean, in human, we don't work on the patients, okay? In a mouse model, after treatment with this, you know, the stress, chronic stress, we could look at that process of what is going on in the synapse density, the synaptic strength, and so on, in this area of a pathway where engram cells are there, and how the positive engram activation will restore that. So that's doable, but not in human.
Second part, real quick. So you showed that it lasts for an extended period of time. If you continually activate that, will it slowly go away over time because you're not pairing it with any other type of sensory activation?
So right now, as I told you, for the non-acute effect, you know, we tested only 2 days, okay? And then if you look at the 2 days later, after multiple rounds of activation with the light, the restoration is 100%. So we could look at the longer period, and this is ongoing, okay? Now, there has to be some kind of plasticity involved in this process. So we will see how long this effect will last. But also, we will try to find out whether how plasticity, synaptic plasticity or connectivity of neurons changes, engram cell changes. Yeah.
Is there been any understanding of how these engram cells, whether their densities or affect, especially in Alzheimer's or dementia-related diseases? Have you or others looked into that specifically?
Yeah, that's a very good question. And So I just want to, I hope my answer will not be too long. I tend to talk too long. Dementia, Alzheimer, it's known in the human that there is a period of maybe a year or longer where the low plaques can be found obviously in any part of the brain, and yet animal is already, not animal, patients are already amnesia. They cannot form a memory or they cannot recall memory, okay? There are similar kind of, one can simulate this in mouse by genetic transgenic animals. And it looks like in the early phase of mouse Alzheimer model, there is a period where the situation is very similar to the one I described, which is retrograde amnesia. In other words, animal cannot express memory, but actually memory is there, because if we go directly to the engram cells, then the recall is restored. Okay? So I think this is a very interesting situation where most people talk about, memory research put a lot of emphasis in ability of impairment in making memory, storing information from experience. But actually, many cases of amnesia, I think it's not necessarily due to the inability to store memory information, but inability to effectively retrieve memory information. I think there will be more and more other cases will show up like that, and both in animal and probably also in human. Did I put this in? All right. So.
I'm Donald Bruce.
Sorry.
I work on ethical questions in technology, and this is not a field that I have any experience, so forgive me if I ask a slightly naive question. But you said this is mouse, not human. How readily could you translate your findings as far as they are into the human understanding, given that we are not the same animal? I mean, is this something which you can truly say, this is a model of humans? Okay. Or is it really just, here is a very good way of probing the basic mechanism in a scientific way, right, but we would have to say, whether humans are the same, we don't know.
I assume what you're asking is how one can translate this finding in a mouse to human system. Is that right? That's the main thing, right? The mechanism. So my answer is, of course, we don't know, okay? But at this fundamental level of information storage for memory, I think we share very, as far as the fundamental mechanism is concerned, we share it. We mean the mouse and the human, or other mammals, okay? So in some detail, I'm sure there will be a difference. But for the purpose of this concept of perhaps manipulating the engram-bearing cell, memory information-bearing cell revealed by animal studies, at that level of fundamental mechanism, I would like to think that that would apply also in some at least some type of memory in human, like episodic memory. Okay. So we'll see, how this will go, yeah.
Thank you for the presentation.
Is this on?
You have identified neurons and their connections as the basis of memory. Now, this is like in a language where you have a dictionary containing all the possible words, which you then combine into phrases and meanings. So my question is, so a neuron and the connectivities define the memory. So this is like in a language where you have words like the atoms of meaning, which are then connected in phrases.
Right.
And this means you can estimate probably the number of words in the dictionary of our brain. How many different memory words, neuron cells, do we have
Right.
to combine different
Yeah.
phrases in our memory?
Of course, you can moderate and calculate, but just before doing that, I want to remind you that this is the combinatorial connections. It's not one set of cells connected to another set of cells, and for particular memory, and for different memory, different set is connected. It's overlapping things. So it's a pattern, one should say pattern of connectivity. Okay. So there are tremendous repertoire, if you think about it. If let's say about 2,000 cells in the dentate gyrus is involved for this the connectivity. If you have another 2,000 cells which is overlapping by 1,000 with the first one, that will show the different connectivity, different pattern of connectivity. So they may be able they may be able to encode different memory of similar type.
So how many of these memory cells does a mouse have and does a human
Right
have?
now, the way we have an upper estimate, we don't have the minimal requirement. In a dentate gyrus mouse, there are 1 million cells, granule cells, and we are activating about 3% of those. Okay? So that's the 30,000, and the 30,000 cell is activated, and reactivating those cells is sufficient for animal to recall memory. Okay? That doesn't mean 30,000 all are needed. Okay? So that minimal estimate of how many cells actually reactivation is sufficient for behavior memory recall, that experimental determination has not been done. But it's that kind of order. They are probably like 5,000, 10,000 cells out of 1 million for specific memory. Yeah. And I want to emphasize it's a combinatorial connectivity that counts specificity of a memory.
Yes.
Okay. It's not just a linearly additive. Yeah.
Thank you very much as well. Is there the contrary also possible that...
I'm sorry.
Is the contrary also possible? I mean, memory is associated as to be good, but sometimes with traumatic events, we always forget actively things, and we have to suppress things.
Right.
You think you can also look into your model for how do we forget things?
Okay. That's a very good question, and it's a very hot subject now in the memory research. Do we have active mechanism to get rid of memory, okay? And rather than memory cells just fading passively, okay? And there are some claims that, that such active process exists, mostly so far studied with the Drosophila, so invertebrates, okay? But people are working on now mammal with a mouse, and I'm not surprised. I won't be surprised if there is a process like that. Okay, you think about this. If you this good memory and the bad memory, the enjoyable experience memory and the memory of the scary experience. If these cells, as I said, in terms of connectivity of circuit, the competing, I won't be surprised that we have evolved, fortunately, to suppress or get rid of a certain type of memory. Okay? So we'll see. But as I said, it's a very active field of research now being really investigated using all these technology to see whether to establish there is an active process of memory forgetting.
But the question is, will it be irreversibly suppressed?
Well, we don't. Yeah, we don't. Well, many cases that when you have amnesia, it's not necessarily completely gone, like Alzheimer, okay? When Alzheimer advances, then the cells are dead, so irreversibly gone, okay? But in the early phase, as I told you, animal show or human shows the memory deficit, but we demonstrated, at least in mouse, memory is still there, okay? So the fact that you can't, the animal or human cannot recall memory doesn't necessarily mean the memory is gone. Okay? So it's the same, probably same thing is happening with the forgetting process also. In some cases, maybe cell is destroyed, okay? And therefore you completely forget. But in other cases, it could be more regulatory process, particularly maybe early phase of forgetting, and therefore memory could still be there, but it's suppressed. So that's the way we would think.
Thank you very much for this talk. I was thinking, could it be that there are a kind of hierarchy in the way that the information is stored and retrieved? So I could imagine that it's like having a database where all these engram cells store the information, but you have another level that is the software that is able to activate and then recall the information. And if this is not functioning well, is where you may have this kind of lack of memory, or you will come. So could you make an experiment?
I see your point. Could you ask me one more time?
If you will make an experiment with providing two different training methods,
I see.
Will you have a kind of
Very good question. Very good question. We would love to do experiment like that in order to show specificity of manipulation. Right now, technically, this is not possible, okay? You need two kinds of, for instance, channel rhodopsin would be activated with a different light of different wavelengths. And people like, Karl Dijsselhoff and these people are working on it, but what they have is not good enough. It's a separation of, it's not good enough. So it's not worth trying with the tool they have now. But I think eventually they will find their tools like that. Yeah, that would be very nice if one can do that. Yeah.
Is there an opportunity for computer simulations to complement your experimental work?
That is being done. I skipped many slides here. So in optogenetics, you can manipulate both ways, okay? Activating them to let animals recall memory, or inactivating them so that they won't show the memory. Or you can also do this in order to understand the pathway of memory information flow, okay? So area A and area B in the brain, if A is upstream of B in a flow of information, okay, even if you activate A, if you inactivate at the same time B, which is downstream, then you do not get the memory. So you can order, in fact, you can order the area of the brain which is crucial for certain memory reactivation by combining this, trying the different combination, 2x2 combination of a manipulation with a positive opsin and a negative opsin. So this is exactly what is done in one of the slides I skipped. So yeah, answer is yes, one can do that. Okay. Yes. I can't. Yeah.
Just a quick comment from the perspective of in government when one has to think, is it specific for mice, Drosophila, or human beings? Actually, the medics time and again make observations which show that memory retrieval, as you describe it, is exactly the same in humans. For example, in neurology, we know cachosmia. Well, my pronunciation is bad, but it's after a trauma to the ethmoid part of the brain where the olfactory receptors go
Yes.
up. If you've had damage at that stage, so that they have to regrow their neurites, the patients describe for a while that their smelling is completely confused, especially complex memories, raspberries.
Right.
Forest will all of a sudden smell of cassis. Nothing except cassis. Don't smell mushrooms. Women will smell of lavatories. It's terrible. I mean, your poor mice, I mean, and their challenge, I mean, I had a lot of sympathy from a human medical point of view. But what is interesting and what actually corroborates your observations in mice is that these same patients, within days, can recover their memory, and all of a sudden, there is lovely mushroom smells in the forest. Cassie will be restricted to the fruit, and so on. And girls, of course, no problem at all. So what I'm trying to say is that there must be ways of finding out if memory can be retrieved, even when actually the patient does not have access. And there are situations, of course, when the cells are dead, well, then it's tough.
So you know this, there is something called psychotherapy. I'm sure some of you are psychotherapists here. And they are actually, for the patients, the depression case, the patient which do not respond to all the available medicine, what the psychotherapist does is to patiently talk with with a specific patient and try to make them reminded of the positive experience they have had before they fell into depression. Okay, and apparently, to some extent, this works. In other words, without using this artificial method of light-induced activation of artificially labeled engram cells, actually it's being done with using the power of a natural recall cues. Except it looks like in a depression patient, the ability to recall the positive memory, enjoyable memory, is apparently suppressed as a result of falling into depression. Okay, so that's what the therapists have been doing, and in fact, our data with the mouse data, but gave the scientific justification for the psychotherapy.
See, the question with the cachosmia is insofar interesting is that when it comes to people who are colorblind, it's very easy to imagine the physics of color blindness, whereas olfactory system is a few thousand different receptors, and there's many allelic variants, so we cannot, by reductionistic logic, exchange our experiences in terms of useful memory, which we can elaborate on. And the interesting thing with these patients with cachosmia is that they have to do a work which is instinctive. And for a long time, it's a disaster because the women smell of the lavatories and so on. But then all of a sudden, from one day to the other, they can retrieve their memory. It is as if all the mosaics fall into place.
Well, maybe we can discuss that. Yeah.
Excuse me.
I think
I have a question.
Okay.
Yes, doctor. I apply the principles of chaos theory to electrocardiograms. But in doing research, I found some very interesting experiences regarding electroencephalograms and, for instance, the use of marijuana in patients. We found that, as you know, the normal electroencephalogram is very irregular, has irregular patterns. And when subjects were subjected to marijuana, the electroencephalograms became regular patterns, and in the process, people lost memory. Have you seen any experiences in working with memories and drug addicts and losing memory? And so, because what we found in And it has a lot to do with nano, because the electrocardiograms of a regular healthy subject were macroscopically, they were very regular, as you know. As you go to arrhythmic states, it becomes macroscopically more irregularly, and to fibrillation is very, very irregular. This is what cardiologists see, but when you apply chaos theory at the micro level, the healthy electrocardiogram is more chaotic, and the fibrillation is ordered. So the question is, how can we explain that regarding electroencephalograms?
Well, that I cannot explain. Well, the only thing we can say is that the fact that based on our results, when you animal or human show, particularly human, show some amnesia, inability to recall memory, that doesn't necessarily mean, okay, one should not interpret this as a result of the loss of memory. The memory is often there. So it's a matter of inability for the recall cue to have access to the cell populations or circuit which is holding a memory information. So I think in human, to translate what we find in human directly is a big challenge. So I cannot tell you a very specific interpretation of what you are finding there, but hopefully, work like this will continue to give us more detailed information, and therefore may become more applicable in the interpretation of human results, human patient results. That's all I can say at this moment.
Susumu-san, I think over lunchtime you will have all the people around you who want to get more information. I thank you very much that you have been here. It is really great. And
thank you very much.